and for the introduction and the invitation to speak in your webinar series. So today I'm going to talk about some work that we published a couple of months ago on the origins of three-dimensionality in drug-like molecules. So a quick overview of the talk. We're going to talk about the motivations for introducing enhanced three-dimensionality into our drug-like molecules, the reasons we want to do this, and investigate options. We're going to look at two descriptors of three-dimensionality, one of which is the principal moments of inertia, which has been used for a number of years in drug discovery, and one that we developed at the ICR a few years ago called the plane of best fit, which I'll describe both of these in a bit more detail. Then what we're going to do is look at three-dimensionality in an extant and exemplified medicinal chemistry drug-like space using ChEMBL as the model here. We'll look at those extant drug-like molecules in total as whole structures, whole parent molecules. We'll then look at using retrosynthetic analysis, using another algorithm developed here by Nick Firth, to pair back the molecules to their constituent building blocks. We'll also look at fragmentation analysis, again to look at the building blocks of the molecules. And then we'll look at enumerating large virtual libraries to see how we can modulate three-dimensionality in our molecules according to the scaffolds we start from. So as I've mentioned already, all of this work has been published in a special issue of Future Medicinal Chemistry, recently edited by Jürgen Bayerat. The paper link is available from the website, and I'll just assure you we've published it open access, so you can freely download it without needing any subscription. The paper was led by myself, but also Joshua Myers, Michael Carter, and Yi Mok in the in silico medicinal chemistry team at the ICR. So looking at moieties, structural moieties that promote three-dimensionality in drug-like molecules, we can quickly classify at least 4 different types of structural moieties that promote three-dimensionality. So there are quaternary centers, here highlighted by Effexor as a blockbuster Pfizer drug. There are spiro ring systems, here represented by Avapro from Sanofi. Bridged cycles, bridged bicycles, Spiriva here from Boehringer, and then also conformational restriction, exemplified here by Zetia at Merck. And these are, you can see these are quite big blockbuster drugs, and looking through the top 200 drugs that are on the market, there's a considerable amount of them that contain these kinds of moieties. So they're definitely of interest in meeting unmet therapeutic need. Additionally, if you look at what we have in, say, a typical fragment library or a screening library, and also vendor collections, what's commercially available, you can see the number of bridged and spiro systems in these screening-like libraries is significantly lower than you'd expect from a ChEMBL medicinal chemistry relevant database. So here we're looking at a few percent or less for a lot of the bridged and spiro systems, but if you look at ChEMBL, it's enriched by 2 to 3-fold in some cases. So it's clear there is enhanced shape diversity in exemplified medicinal chemistry space. But how do we increase this, and how much is it important, and at what stage in a drug discovery project should you be thinking about enriching this three-dimensionality? So our motivation for looking at three-dimensional molecules, there's a few ideas we have in this context here. Looking at mimicking natural products. If you look at natural products, endogenous ligands, they tend to incorporate quite three-dimensional scaffolds. And we need, if we're going to be competitive with those endogenous ligands, we need to reflect the three-dimensionality and the morphology of those binding sites so that we can exploit the interactions in that binding site in an appropriate way. There's also some speculation about improvement in properties. So, for example, it has been experimentally observed that some molecules with more three-dimensionality do convey a more aqueous solubility, which is an important aspect at least to explore and if possible, exploit in our design strategies. And also addressing new and challenging drug targets. So an example here is protein-protein interactions, which is a hot topic of the moment. So that's our motivation for looking at them, the three-dimensional molecules. Another motivation is the recent work from Frank Lovering and colleagues at Wyeth, the Escape from Flatland paper. So this was using a FSP3, so fraction of sp3 centers in a molecule as a measure of three-dimensionality, although it's really a measure of topological complexity. But in this paper, they tried to demonstrate that enhanced three-dimensionality led to improved clinical success. So let's start with the principal moments of inertia. This is a long-standing descriptor used in drug discovery for a number of years. Simply put, it's as you can see on the left-hand side, a ternary plot where at the three apexes of that ternary plot, the triangle, you can see that the shapes are rod-like, so long and thin, disc-like, very flat but round, and sphere-like, so spherical in nature. And then between those points, those apexes of the triangle, you will change your morphology of what you're describing. So you can see that if we go halfway down that flat axis, you can see the azindole there as a flat molecule, but it's got rod-like character as well, because it's about a fused bicycle. And then you can see on the right-hand side of the triangle, you've got a cyclohexane that's got more three-dimensional character, but not quite spherical, as you'd see with the adamantane ring in the top right. So the principal moments of inertia, or PMI, is quite intuitive in presentation, but we must remember it's also size independent. So as you can see there, the Earth is spherical, but so is a golf ball, and these will be identically spherical, even though they're vastly different in size. So this should be considered when you're using this algorithm. On the right-hand side, you can see a plot of the ChEMBL drug-like small molecule data set that we used in our analysis. This is a density ternary plot where the colors represent how many points were within that particular ternary or triangle cell. The legend on the right-hand side tells you about that. So what this represents, I think quite clearly, is that most of ChEMBL drug-like small molecule space is heavily represented by things that tend towards planarity. However, there are quite clearly a number of structures that enrich that three-dimensionality of those molecules, but you can see there's a heavy balance towards the more planar molecules. And it's not really clear how three-dimensional we have to be. It's probably somewhere pushing of that three-dimensional envelope, but not necessarily too far. The other descriptor we've used in this study is the plane of best fit. So this was described by Nicholas Firth, a former PhD student in the research group here, where it's the best and probably simplest way to put it is extending the line of best fit in a two-dimensional scatter plot to the plane of best fit in the 3 dimensions. So you're trying to find a plane that minimizes the errors of each heavy atom in a particular molecule, so that plane best fits the data. And then you can average the distances of each of those heavy atoms from the plane. So quite clearly, if you found the plane of best fit, and it's quite a flat molecule, that plane of best fit will have very low errors from the plane to the heavy atoms. However, it's more three-dimensional, like the example there with in particular the sulfonamide moiety. You can see that kink in the conformation generates and enriches three-dimensionality in the final molecule. So we believe this is simple in concept and an interesting descriptor to use in conjunction with the principal moments of inertia. Additionally, it has a degree of size dependency as well, which is a very important aspect to consider when used with PBF. Also, it's an unbound descriptor, so theoretically it runs from 0, meaning entirely flat, to infinity, whatever that means in the context of drugs. We're not sure, but the molecules we've at least looked at, they don't go anywhere above 2 or 3 in terms of the deviation from the plane. So here's some examples, further examples of the plane of best fit with structures from ChEMBL. So you start from the left-hand side, you can see both the 2D and the 3D structures. You can see as you move along the continuum of the PBF descriptor, that the molecules and the conformations become much more three-dimensional. So again, you can see on the right-hand side at the top, there's a clear kink in the conformation that's introduced by the sulfonamide moiety that's generating a much more three-dimensional molecule than you'd expect without that moiety. And what we found in the study we published in 2012, this was in Journal of Chemical Information and Modeling, and I believe it's open access as well, is that PMI and PBF actually work quite well together. We're not suggesting that you just use one or the other. It's quite good to look at your data in lots of different ways if you've got relevant descriptors. In particular, where we found PBF to be most importantly used is when you're looking at just moving a little bit away from that flat edge of the ternary plot. It tended to be much more discriminatory in our analyses than PBF. So you could tease out more than PMI. Sorry. So you could tease out more interesting three-dimensional character at that three-dimensional structural level. Then what we did is we looked at pairing back the molecules using a scaffold tree fragmentation approach that was published by Ansgar Schuffenhauer and colleagues at Novartis around about 10 years ago now, and we followed up with some additional work at the ICR 4 years ago. This is a retrosynthetic approach where you pair back ring systems, starting with the parent molecule, and then moving to, in this case, the level 3 molecule, which is essentially the definition of a Merkel scaffold from Bemis and Merkel's work. And then you iteratively pair back the ring systems of the molecule until you're left with one ring system. So again, these are fairly simple and intuitive rules that mimic some type of chemist thinking, and it allows for a large-scale, systematic, objective and invariant evaluation of drug-like chemistry space in terms of the building blocks. So I think here, Carsten's going to pause for a quick poll.
Yes, this will launch our only poll in this webinar, and we would like to know who we are dealing with as an audience. So if you could please vote here whether you are a computational chemist, a medicinal chemist, biologist, crystallographer, or none of the above. And I can see that there's already quite a few that have voted. I'll give this a few more seconds here. Seems to be a clear winner. I'll give you a little more time and I will close the poll in 5, 4, 3, 2, 1. So we have a majority of computational chemists here, well, quite almost a majority of computational chemists, closely followed by medicinal chemists. That's great. A few biologists, even fewer crystallographer, and some are probably physicists or just interested in what we have to say. Okay, thank you for that, and I am switching back to Nathan's presentation. Nathan?
Yep, thanks, Carsten. So hopefully you can still see my slides. So now, using the scaffold tree analysis I've just briefly introduced before the poll, we can use those same density ternary plots I presented with the entire ChEMBL library, but according to the level of the scaffold tree of those molecules at which they've been pruned. So this is sort of returning to Flatland as opposed to escaping from Flatland, because I should expect the molecules will become more flat as you pair back the ring systems. So looking at level 5, you can see there's some heavy three-dimensional molecules in there, and you can still see those in levels 4 and 3. And then as you go into 3 to 2, you can see a reduction, particularly in the density, maybe not the coverage, but the actual density of the molecules, really going back to that flat edge, which is very marked in level 1 and level 0. And in level 0, obviously, you would see down the bottom apex of the triangle, the ternary plot, that things are much more disk-like, which is, as you'd expect, from many ring systems. So this I think shows that a lot of our medicinal chemistry relevant scaffolds tend towards the planarity rather than the three-dimensionality. As you pair back the complexity of a molecule, you'd expect there to be a greater tendency towards flat things rather than 3D things. But how can we use this in our later designs? So similarly, if we look at the plane of best fit in a scaffold tree analysis, it might be more clear to see it in this way. As you pair back these levels, and not every molecule will have the same number of levels, that's why this one goes up to 8 in this case, with a few pruned out. You can see at that level 2 to level 1 stage there's a drastic reduction in the three-dimensionality of the molecules, but you can see that trend running pretty much from level 6 down to 0. So clearly this is increasing more planarity as you reduce the size of your molecules, which is, I think, not that surprising. But it's important to think about how we use this. Another way of looking at this is looking at what we term the level 1 scaffolds, which is based on the work from Sarah Langdon in the group some years ago, taking the level 1 scaffold, which means that you'll have 2 ring systems in your molecules, taking all molecules that contain that level 1 scaffold in ChEMBL, and then mapping on not just the scaffold itself, but the exemplified parent molecules that exist in ChEMBL. So here we've picked up on the top 8 most occurring scaffolds in set 3-dimensional bins according to the planar best fit. So starting on the left-hand side there with a flat fused aromatic ring system, moving up to the larger, the G and H cores, the larger compounds with the sulfonamide moiety in them. And then below you can see the density plots, sorry, not the density plots, the scatter plots, the ternary scatter plots of the black stars that represent where the scaffold lies in the PMI plots, and the red dots where the exemplified molecules lie. So let's look at 2 of those in particular, the flattest and the most 3-dimensional that we explored. You can see that the parent molecules, the molecules that have been actually synthesized and reported in the medchem literature, explore quite a good degree of 3-dimensionality in the parent molecules, and always more 3-dimensional than the original, or in most cases, the original scaffold. Whereas you look on the right-hand side, the biphenyl with the sulfonamide moiety is, in the majority of cases, the 3-dimensionality reduces. So adding anything to those scaffolds is going to lead you to a reduction in 3-dimensionality. So I guess one speculation, and maybe not a total conclusion here, is that we can modulate 3-dimensionality easier with more planar scaffolds and with more 3-dimensional scaffolds. However, if you look at it on the planar best fit, you can see with the box and whisker plots in the bottom left, using the same cores, we can pretty much explore a similar kind of 3-dimensionality in all of those scaffolds, regardless of the original 3-dimensionality of the scaffold. So this suggests that perhaps those more 3-dimensional scaffolds do not necessarily translate into more 3-dimensional structures, but that also means that the more planar scaffolds would not also enrich on that. But then you have to start wondering about your computational, your chemical complexity, and also the structural moieties you're using if you really want to introduce a sulfonamide group, and if that's going to help you in all of your designs. So another way of looking at this is using SynDiR, which is an algorithm we published last year in our Morph paper, which is an open access paper in the Journal of Chemical Information Modeling. Here we have 2 plots where we took all of the ChEMBL set. We applied the SynDiR rules, which fragments up similarly to recap and other retrosynthetic rules, fragments up the molecules in ChEMBL, and then we look at the occurrence of those fragments in the exemplified medicinal chemistry space. So on the left-hand side, we have the actual occurrence of those moieties. So that means not just that they occurred, but they occurred in how many instances of a molecule reported in ChEMBL. So you can see here with the red bounding box that very many things that are used are flat as building blocks, much higher than the 3-dimensional aspects of the PMI plot. But if you look at the unique occurrence, so this is where a fragment's used, but we don't count how many times it's used, you can see that there's an enrichment of 3-dimensionality. So there's clearly, there are moieties being used that are more 3-dimensional, but they're not being used as often as the more planar substructures. So again, most substructures that are used and also exist tend towards planarity. So this again adds some credence to the idea that 3-dimensionality may originate, or at least may be modulated by using these planar moieties in concert to emerge 3-dimensionality in the parent compounds. Again, looking at this data set of the SynDiR-generated ChEMBL fragments, but this time using the planar best fit as the descriptor. So on this plot, we have, again, it's a density plot, but here we have on the x-axis the log with the frequency. So the more often a structure is used, the further along the x-axis it's going to be. And then on the y-axis, we've got the planar best fit. So the higher up that y-axis, the more 3-dimensional the compounds are going to be. And within each cell, the density of exemplified substructures in that cell is represented. So the more red the cell, the more individual substructures are represented in that cell. So again, what you can see, the red bounding box shows that there are three-dimensional things going up to 1 in the PBF descriptor, but these aren't used that often. Whereas it's more those things on the edge of the green and the blues, where these are the more three-dimensional things that we should be considering perhaps in our designs. So again, most substructures tending towards planarity, and we should be looking at these planar moieties and how we can use them together for three-dimensionality to emerge. The next step in the analysis, so I'll just summarize what we've done so far. We first looked at the ChEMBL structures themselves, the actual parent molecules. We've looked at pairing back the molecules down to their scaffolds, incrementally removing the ring systems until we get to the fundamental ring system of the molecule. We've looked at using a retrosynthetic approach to see where the theoretical chemical building blocks are coming from in terms of their PMI and PBF distributions. Now we can take, again, going back to those most frequent scaffolds in each of the PBF bins we defined, we can take those scaffolds, and now we can take also the most common monomers we identified using the SynDiR algorithm for fragmentation, and then enumerate virtual libraries using these scaffolds with those monomers. And again, along the bottom, you can see the ternary plots, the scattered ternary plots, where the black star indicates the three-dimensionality of the level 1 scaffold, and the red dots give you the objective enumeration of virtual libraries using those most common structural monomers that we use in the analysis. So looking at this a little bit more clearly, you can see that there is a greater increase in three-dimensionality when starting flatter scaffolds, but that's really only in the distribution. If you look at the box and whisker plot at the bottom, so you can modulate using flat scaffolds, but in this case you can see that we can, when starting from more three-dimensional scaffolds, you can increase your three-dimensionality as well. So that's the virtual enumeration libraries. So at this point, it's probably coming to the end. What we've done here is we've presented approaches to analyzing three-dimensionality. So we've looked at exemplified medicinal chemistry space. So this is what has existed, has been made, has been reported. Pairing those back to the scaffolds from which they were made. Fragmenting into constituent substructures using a retrosynthetic analysis that is objective and invariant and mimics to some extent the medicinal chemistry approaches. And then enumerating the virtual libraries, generating theoretical libraries that have as many of the variances removed as we saw in the original parent molecules that have actually been synthesized. This is what could be synthesized from the same building blocks, and seeing how the three-dimensionality can be modulated. So this has given us some ideas about when and where we can enrich three-dimensionality if it's needed in a drug discovery project. So it's clear, I think, that constituent substructures that are used in drug discovery programs are relatively planar. Also, looking at the medicinal chemistry relevant scaffolds according to the level 1 scaffold of the scaffold tree are also typically flat. So the things from which we're building tend towards planarity again. However, this is where three-dimensionality can be modulated. It's in the design of the parent molecules by maybe combining planar scaffolds with planar moieties, but bringing back to the original slide on where the structural moieties enrich three-dimensionality, things like restricted conformation, exit vector diversity, sulfonamide groups, these kinds of typical tricks. And I think it's fairly clear that we've shown that it's not that important to have inherent three-dimensionality in your scaffolds and your groups. And this is particularly interesting in your scaffolds, because if you look at fragment libraries, which are quite small, it's maybe a good idea to not increase your molecular complexity too early in a drug discovery program, because that might lead to additional challenges later on. But you can modulate that three-dimensionality when needed in that project, as and when the need arises. So with that, I'd just like to acknowledge the members of my group here, photographed in various social gatherings. In particular, the co-authors of the study, Michael Carter, Joshua Myers, and Yi Mok, but also the other past and present members of the in silico group here, Nick Firth in particular for his work on the SynDiR algorithm and the planar best fit algorithm, and Sarah Langdon for her work on the Scaffold Tree analysis and looking at the scaffold diversity of exemplified medicinal chemistry space. I'd also like to thank Julian Black, who's our head of chemistry at the ICR, for his interest and ongoing support for our efforts in analyzing computational chemistry at the ICR. With that, I'd like to close and thank you for your attention and ask for any questions you may have.
Thank you very much, Nathan, for this really exciting talk. There are a few questions. Let me see here. The first question is, in all these examples, you discuss three-dimensionality for 3D compounds and scaffolds that were probably calculated. How exactly did you calculate those, and how do the results depend on the conformation generation process?
It's a very important question. So this is mentioned in the paper, but there wasn't enough time in the slides to go through this. So we use a single conformer generated from the Carina program from Johnny Gasteiger's lab, which reflects with low error the most likely small molecule crystal conformation. Now, obviously, conformational flexibility is going to change the analysis in some cases, but we made it clear in the paper that we're not going to challenge or investigate the conformation problem in this paper, but that's some follow-on work we're exploring now.
Okay. I also have a question. You mentioned that most of the medicinal chemistry scaffolds are flat. How do you think the success of a compound would be influenced by modifying that? So, for example, replacing a flat scaffold by a more three-dimensional one.
So it really depends primarily on the morphology of the binding site you're trying to target, and if it will allow some more enhanced three-dimensionality in the core scaffolding. So that core scaffolding can be enrichment of not only the three-dimensionality of the scaffold itself, but the three-dimensionality of the exit vectors you can diversify from. Once you've got an optimal binding or binding from which you can work for a drug discovery project, then you might want to be thinking about other properties you can optimize. So that's potentially where you might want a more three-dimensional scaffold for optimizing other properties as you go further down into lead and lead opt. So I think it's an open question, really. It's difficult for us to quantify that.
Maybe this also tells us a story about what kind of binding sites we're dealing with, because if we have rather flat scaffolds, this means in that area at least we're dealing with more lipophilicity and also possibly with less space, right? Because as you said, the exit vectors we're working from this scaffold are rather in the same plane rather than pointing into all different kinds of directions.
Yeah, for sure. And also kinase inhibitors, there'll be lots of those in ChEMBL have been reported over the past 10, 15 years. So that's something we could do, is look at from where these molecules have been abstracted and see if there's a common target classes being investigated, and there's maybe a propensity for more planar scaffolds when you're targeting the rather planar donor-acceptor-donor motif in a kinase structure.
Right. Another question is, have you performed the same analysis on natural product data sets? Or a data set?
We haven't yet in quite the same way as we've done here. We have done some work on looking at three-dimensionality of endogenous ligands, but none of that's been reported in this paper. So we have looked at it, and it might be interesting to see how that is reflected in the three-dimensionality concordance with natural product space, because quite often that's what we're trying to compete with.
Okay, I think that's it in terms of questions. If there are not more questions, then I would like to thank our speaker from today again, Dr. Nathan Brown. Thanks very much for this exciting talk. We will see you back at November 23rd, 3 p.m. Central European Time. And up until then, thanks very much for your interest in this. And I will close the webinar for today then. Thanks again to everyone.